And this time she saw some really interesting stuff. That in wounded eyes, 48 hours after injury, suddenly the PAX6 mutant stem cells were awake, wild type. And as I've described, if you injure one, after 48 hours, the stem cells wake up in the other eye and start proliferating. And it was the same for PAX6 mutants. So in the contralateral unwounded eyes, after 48 hours, again, nearly all the PAX6 mutant limbal stem cells were dividing. So what we're seeing here is that there is a problem with PAX6 mutant limbal stem cells, but it's a delay in the response to wounding. It's not a failure of response to wounding. That PAX6 mutants do have stem cells. These stem cells can work, but they take longer to respond to wounding. And Akhlam's little cartoon is now this. That here's the wild type situation. This is what I've already showed you. The PAX6 mutant mice with the smaller eyes, the only difference is at 24 hours. That in the wild types, the injured eye stem cells have activated, and in the PAX6 mutant they haven't. But after 48 hours, there's no difference. The PAX6 mutant limbal stem cells can activate pretty much as well as wild type. That's a very subtle defect in the stem cells. And the question is, can that explain the clinical phenotype? That this very mild deficiency of limbal stem cell, is that enough to explain the catastrophic failure of corneas that we see in aniridia patients? In order to answer that question, we have to look at anything else that might be happening in the aniridic cornea. And another PhD student of mine, Jingxing Oh, really about 10 years ago now, was able to investigate that question. What goes wrong with, not with the stem cells, but with the actual corneal epithelium cells themselves, the central corneal cells. And one of his papers is up here. I mean, what he was able to show is that the PAX6 mutant corneal epithelium is a bit of a mess, and it's fragile and it's lost its barrier function. So well, let's start with the nice pictures on the right here. This is a wild type mouse eye. This is a PAX6 mutant wild type PAX6 mutant eye, and you can see the opacity forming in the cornea. This opacity affects both the epithelium and the underlying stroma, and it's associated with a function of the mutant cornea. This is some scanning electron microscopy of mouse corneas, and the wild type corneas have a nice smooth barrier surface. The mutants, unfortunately, are not like that. Their epithelium is full of holes, and there are cells escaping from and flaking off from the surface of the mutant. Because the cornea is full of holes, it loses barrier function. If you drop fluorescein on a mouse eye, normally it should stain only the very upper surface of the eye. If you drop fluorescein on a PAX6 mutant eye, you can see the fluorescein penetrates the whole cornea. So these corneas have lost their barrier function. Ooh, I'm getting some feedback now. What does that mean? There is getting into the PAX6 mutant epithelium. That predisposes them to higher levels of oxidative stress, and we've measured that directly and shown that the PAX6 mutant corneas are under oxidative stress. What the stress does is it causes the actual red color, the green color here is oxidative stress, and And the red colour, the knife edge, and any small thing going wrong, such as holes in the epithelium, loss of protective enzymes, appears to be able to tip the cornea over into a pathology. Why this affects PAX6 particularly wasn't immediately obvious, but Jingxing showed something else, which is that PAX6 itself is susceptible to oxidative stress. And what it's susceptible to is that oxidative stress drives it from the nucleus. So in a PAC, this here is a normal wild type cornea, and PAX6 in green should be expressed in the nucleus. The protein should be localized to the nuclei of the epithelial cells. In a PAX6 mutant, it's patchwork. Sometimes it's nuclear, but in other cells you find it's gone cytoplasmic. And Jingxing was able to show that is a response to oxidative stress. Because if you take normal wild type cells and put a low concentration of hydrogen peroxide on them, the PAX6 leaves the nucleus. You can see how that can exacerbate the PAX6 phenotype. Because if cells have a little bit less PAX6 than normal, that leads to oxidative stress, which drives the PAX6 that there is out of the nucleus. And remember, PAX6 is a transcription factor. It has to be in the nucleus to work. So what we have labeled this, because we're not serious, is the triple whammy model of aniridia-associated keratopathy pathology. That the aniridic corneal epithelium is fragile and it's got holes in it and it loses barrier function. In combination, it expresses lower levels of stress-responsive enzymes than it should do. And together what that does is that it leads to increased oxidative stress, which in turn expels PAX6 from the nucleus. And this leads in the PAX6 mutant to a chronic wound healing state and dedifferentiation. And the limbal stem cells respond to that by slightly increasing their proliferation, even in the apparently uninjured aniridic eye. And then this chronic wound healing state appears to delay the ability of the stem cells to respond to any subsequent acute wound. Hope for the future is that if we add reducing agents to PAX6 mutant cells, things like, beta-mercaptethanol, or a much more friendly reducing agent, glutathione, then we can restore some of these properties. But if we add reducing agent eye drops to PAX6 mutant mice, we can start to restore, we can push PAX6 back into the nucleus, and we can start to restore some of the wound healing. So this figure here is showing that adding reducing agents to PAX6 mutant cells restores their wound healing ability pretty much to wild type levels. These are control cells, cells with hydrogen peroxide where everything gets worse, cells with beta-mercaptethanol and glutathione where everything gets better. And this is a control dimer of glutathione that shouldn't have any effect, and thankfully it doesn't. Now, okay, Jingxing produced this summary diagram of how PAX6 deficiency causes opacity. And yeah, I laughed when I saw this. It's like watching A Beautiful Mind. I mean, this isn't a summary diagram. This is a conspiracy theory. It reminds, although this meme wasn't around at the time, it reminded me of this. But what he's trying to show is that the PAX6 deficiency leads to oxidative stress, which leads to a chronic wound state in the PAX6 mutant corneas, which leads to dedifferentiation, loss of epithelial transparency, and opacity of the corneas. And that what we are hoping, our current work, which if I'm invited to speak again in the future, I'll be able to describe to you. Our current work is trying to push this diagram backwards, to start with the opacity, to reduce the oxidative stress, to calm down the mutant eyes, and restore the effects of the PAX6 deficiency such that the eye can survive. And the goal of our work is to create eye drop therapies such that when a baby is born with aniridia, it could start taking pharmacological eye drops that will prevent the corneal opacity from ever occurring, and therefore could potentially prevent the baby from ever going blind. That's all I'm going to say today. So it's been a little bit longer than I expected, but I did mess up the start, I guess. The story about what I wanted to say was that it's been now 32 years since the identification of PAX6 as the aniridia gene. And even now, large areas of the genotype-phenotype relationship are still unsolved. That's good for the young people out there, because there'll be work to do for the next 25 years in researching this and other genetic conditions. But what we have finally identified is that aniridic associated keratopathy is caused by a combination of oxidative stress in the cornea, and it's not primarily a limbal stem cell deficiency, although there is a minor deficiency of the stem cells which prevents them from responding properly to the chronic wound state in the cornea. Mutant limbal stem cells are slow to respond to wounding, but the good news is that they can do it, and if we give them a helping hand, they may be able to do it well enough in order to maintain the cornea during normal life. On the right here, this is my lab. They're all very shy. They didn't want their pictures. But I mean, the people, so Kaya and Eileen down in the bottom left here are the two who are doing the therapeutics on PAX6 mutant eyes to try and restore vision. And Jakob hiding at the back there is currently working on limbal stem cell activity. And Ahlam Alamri was the person who did the wound healing experiments on PAX6 mutant corneas. Everybody else, when they talks at conferences, shows a picture of how beautiful the place is that they came from. I unfortunately can't do that because I'm from Aberdeen. This grey pancake here, the so-called granite city, all the houses are built of granite, and that's kind of what it looks like. But it is, sometimes the sun does shine, and sometimes it is a nice place to work. So thank you for being patient with me. That's all I've got to say. And if anybody's got any questions or any comments on what I've said, I'll be very happy to answer them. I'm going to stop sharing my screen now. Okay. Mm-hmm. I'm very happy to answer any questions. Yes. Anybody there? All right. Okay. Okay. So the question: what the white ring-like structure seen around the cornea as we age? So I don't know exactly the answer to this question. The limbal stem cells are a long-lived cell, and it's in the interest of the eye to prevent them from picking up mutations. So, which is difficult if you are an ocular surface cell, because you're exposed to light, including ultraviolet light, all the time, and potentially other environmental pathogens, mutagens. And so the stem cells have to be protected some way from mutation. And in humans, the way there's two mechanisms that do that. Well, three, but first of all, the limbal niche is pigmented, so that the kind of grey color of the limbal ring is pigment, which helps to protect the stem cells from ambient light and therefore reduces the amount of mutation. But also they're hidden away in specialized structures called the palisades of Vogt, which are little wrinkly projections of epithelium where the stem cells hide away at the bottom. And as we get older, these tend to get thicker and more wrinkly. It's a very slow process, but it does happen. And I think that thickening of the actual limbal niche, together with the opacity and the pigmentation of it, which protects stem cells from light, I think on the whole, that can be responsible for making the limbal ring more obvious as we get older. When one eye is injured, why do we also have activation of stem cells in another eye? That's the essay question, isn't it? I mean, who knows? What we think. Well, there's two answers here: mechanistic and evolutionary. If in the wild, how does this evolve? And acute corneal injury will usually happen in the wild in mammals if they are living in an abrasive environment, sandy, woody, a high matrix grass environment where one eye is subject to abrasion. And there's a good chance that if one eye has been subject to abrasion, the other eye has been subject to abrasion too. So evolutionarily, I can see it might be possible to evolve a scenario whereby the stem cells in one eye activate the stem cells in the other eye, because in the wild, it's not like in the lab with the surgery. In the wild, it's likely that if one eye is being injured, the other eye is being injured too. So I can see why it kind of makes sense. Mechanistically, how it happens, I don't know. When the eye is injured, the cornea, it releases growth factors such as HGF, EGF, KGF, and a number of cytokines that presumably get into the blood system. And maybe the activation of cytokine release from an injured eye travels systemically and reaches the other eye and activates stem cells in that eye too. I'm really only guessing at this. We never did the experiment to test this. And one of the interesting things I think to do, if we'd thought of it, would have been to look at stem cells in other tissues as well. Because if the injured cornea is releasing growth factors and cytokines that activate stem cell activity, maybe they activated stem cells in the skin and the liver and and the gut and everywhere else, and we just didn't look at that. And I think it would be worth. It would certainly answer the question about whether systemic release of growth factors that is causing stem cell activity, if we can show that other stem cells within the circulation have been activated. Ooh, here's another excellent question. Does the stem cells lose efficiency as we age? Yeah. Okay. So I didn't show you this. My first the earlier PhD student, Nada Saga, who looked at stem cell activity in wild type mouse, she did exactly the same experiments, but on old mice. And by old mouse, I mean mice that are over a year old. Remember, most mice in the wild don't live to be a year old. In the lab, 12 months is well past reproductive age, and most mice will be dead by 18 months of age. They can live longer. So Nada did experiments on geriatric mice, and she found that yes, their stem cells in geriatric mice were had showed much lower levels of activation in response to wounding compared to young mice. So if you extrapolate that to people, then we would guess that yes, stem cells do lose efficiency as we age, and that in old people with injured eyes, we would expect lesser of a wound healing response, which fits with our sort of general dogma that old people heal less efficiently than young people. Sorry. That said, there is no our corneas don't fall apart just because we get old. Most old people have corneas that are just as transparent as young people. Just because you get old, your cornea doesn't necessarily go opaque. Lots of old people have perfectly good vision. So if there is a reduction in stem cell efficiency with age, it's not enough to cause degeneration of the cornea. But it may be enough to reduce the wound healing response to injury in older people. Well, thank you for being patient with me, and it's been a real privilege to be able to speak to you today. Oh, that's okay. Ooh, that's very nice. Thank you. That's really lovely. Thank you. That's brilliant. Thank you very much. It's been a privilege to speak to you today. Thank you. Thank you. Okay. Okay, I will turn off my mic and camera, and I hope to see you. I hope to see you again soon. Thank you. Mhm.
Awesome. Thank you, ma'am. Very good evening to all present. It's my immense pleasure to introduce our guest speaker, Dr. Akshay Murugesh, Faculty of Medicine Health, Department of Biotech, Tampere University, Finland. She had completed her Doctor of Philosophy in Marine Biotechnology from Annamalai University in the year of 2015 and a Master of Philosophy in Biotechnology from Bharatdasan University in the year of 2007 and a Master in Zoology and a Bachelor in Zoology in the year of 2006 and 4 from Dock College, Lady Dock College. And she is currently working as a PDM fellow in molecular signaling lab with Tampere University from Finland. And she had experience as an assistant professor and a coordinator for research and development cell and instrument purchase and maintenance. And also she had experience in a research associate and a project trainee. And she's having many funding projects like DST Sub, DBT, and UGC, and also ICMR. They had many publications in the national and international peer-reviewed journals. She has supervised many UG, PG, and PhD students, scholars. She has given many invited lectures and served as an examiner for UG, PG, and MPhil thesis evaluation. Once again, thank you all for giving this opportunity.
Thank you, Ms. Kirtana. Okay. So, whether my slides are visible to everyone? Yeah. So, my presentation is on often purinergic receptor signaling, GPR17, a modulator for treating glioblastoma. So, my lab is focusing research on glioblastoma, and I'll give you a gist of intro of what is glioblastoma. Then I'll focus on the research, various research that's happening on glioblastoma. So this is a very basic intro of what is the brain tumor facts worldwide, as well, especially in Finland. So globally, when you observe, or globally or regionally, or as well in national level, the burden of brain tumor, that is the first graph, the statistics, which is between 1990 to 2016, it says that there is a gradual increase in the burden of brain tumor, and mostly, like the male is very much prone to the tumor than the female, and this might be the fact that of the lifestyle habit. And when you see the next statistics, it says like how much of new cases that comes in every year. Like this is the association between the incidence of the brain tumor and mortality of the brain cancer. Like we call it as HDI, which is Human Development Index. So mostly, like Asians are very much prone to this, which is around 52.6%, and second comes the European countries. Even the Finland comes in this Europe, which is around 22%, and you can see other countries with the less, that is 10 and less than 10%. So you can't see much difference between the new case every year or new case and the death penalty, because the main challenge of this disease is once it has come, or when an individual is getting this disease, it is not curable. So it's very, very challenging disease, and their lifespan is just 1 to 1.5 years. So intense research is going on for the therapy development. So this is again a statistics which is mostly on the Finnish Cancer Registry of 2020, and this compares the number of new cases and year-wise statistics of men and women. And as well, from the graph, you can see the different types of cancers, like breast. I mean, like for women, the breast, the top leading cancer, and for the men is the prostate. In Finland, the prostate is the top leading cancer, and the other cancers, like even the second leads the bladder and urinary tract for the men, and corpus uteri for the women. So likewise, like the statistics, this is exactly the statistics of Finnish, and again, I focus most on the statistics of the Finland, because we mostly concentrate on this particular research of Finland. But the development of therapy is totally for the global use. And this is 5-year data of the brain cancer diagnosis. And almost the red color bar says that around 37.6% of people in Finland, they've been diagnosed with adult. It's between the year of 2000 to 2014, they have been diagnosed with a brain cancer. You can see other countries too, where there's almost like about 20 percentage of the brain cancer incidence was observed. So coming to the basic facts about, the basic classifications of the brain tumor. Basically, the brain tumor is classified as the primary brain tumor and the metastatic brain tumor. Our group focused mainly on the primary brain tumor, and the primary brain tumor is nothing but the tumor which actually been localized. They develop exactly in the brain, different any part of the brain. And whereas the metastatic brain tumor is like it can be in any part of the body, but the cancer cells can move from other parts of the body to the brain, and they can cause all kind of symptoms related to the brain tumors. And coming to the further classifications of the brain tumor, primary, especially the primary brain tumor, which actually we are focusing on. This primary brain tumor is the one where the cells, we call it as glial cells or the gliomas. Those are the cells which becomes cancerous, which are very much similar to glial cells. Glial cells are nothing but, those cells which are present around the nerve cells. So these gliomas, they are very much similar to glial cells, and they develop into tumor. They differentiate very fast, and they develop into tumor. So based on the nature of its differentiation, it's been further classified. The severity also, it's been classified into 4 different types: the low-grade astrocytoma, malignant astrocytoma, glioblastoma, and oligodendroglioma. So of which the glioblastoma, which I have given here as a grade 4, this is a very dangerous irreversible type of cancer, which we actually focus on. Our research actually focus on this particular type of glioblastoma cancer. So according to WHO, those individuals who have this grade 4 type of cancer, which is a glioblastoma multiforme, we call it as GBM. So this type of cancer is very dangerous, and their lifespan is very, very less. And as I said, it's a very simple definition of the glioblastoma. It's a collection of abnormal cells, especially from the brain, or it can also arise from the spine canal. And as I said, the incidents are increasing every year, and their death incidence is like incidence of case of diagnosis is 308, and its death case could be like 1 or 2, and their mean survival rate is it's around 12 to 18 months, right? And coming to the symptoms, the symptoms, as I say, it is in the brain. The brain is the master of our body, which controls all kind of activities, thinking process, and everything, whatever we perform, right? So the symptoms are, they will have an unusual headache, often seizures, memory loss. They'll have some kind of personality disorders, will not be very, very normal to the normal. They won't behave very normal as with respect to the normal individual, and they can't process any kind of information perfectly. They'll have blurred vision problem too, and change in motor control. So almost all the functions, wherever the tumor is localized, and accordingly the symptoms might vary, the localization, based on the localization. So almost all the functions, they deteriorate based on the stage and the severity of the cancer. And how this disease is diagnosed is, we say like gold standard method is MRI scanning is a gold standard method. And in an emergency case, they go for CT scanning. Then, as usual, of biopsy, like removal of samples from the place of tumor formation, and it will be sent for analysis of the biomarkers. Biomarkers which says that it is cancerous cells, and sometimes body fluids also is being used for the diagnosis. So, coming to the treatment strategies, very usual treatment strategy for the cancer: surgery, then chemotherapy, radiotherapy, and this could be of a single type of therapy, or it can be a combination type of therapy based on, as I said, the severity of the disease. And what are the other factors which really lead to the cancer cell formation? I mean, like the features I can say as the hallmark of this particular type of cancer. This cancer, the tissue invasion from one place to another place is very, very high. So they are highly metastatic. For glioblastoma is highly metastatic. That's why their median life or the mean survival rate is very, very less. And their microenvironment will be inflamed, and nothing like in the growth inhibitors, any kind of growth inhibitors will become insensitive, and other factors like sustained angiogenesis or evasion of apoptosis. So these are the hallmarks of this particular type of cancer. So as I said, we were discussing the basics of this, the challenges that the individual or the patient really face for the treatment. Why the median survival rate is very less is because of all these important challenges. And the first important challenge is the blood-brain barrier. I hope like everybody is familiar with this term, blood-brain barrier, because your brain is so protective, it won't allow anything to enter in. So it keeps the organ very safe as a barrier. So when a drug is given, again, this happens, this functions in another way. Like this barrier will not allow the drug to enter into the brain. So, the localization of the tumor, it cannot be targeted by the drug what is given. So the treatment will not be completed. So that is a biggest challenge with respect to glioblastoma. And coming to the tumor heterogeneity, as I said, again, the environment might be based on the localization, the severity changes. The cells or the cancer cells would be of different types of heterogeneous groups of cells would be there. And when you give a drug, one type of cells will respond and another type of cells will not respond to the drug that's given. So targeting all type of cells, a heterogeneous group of cells, is very, very important, even though the drug crossed the blood-brain barrier. And the prognosis is very limited, and we have a lot of communications with the local environment. Like as I said, the communication signaling process would be very, very high. So the process of prognosis is limited, and it hampers the diagnosis process also, and the disease progression is very high. The proliferation becomes very, very high, and progression of the disease is very fast. And all these ultimately lead sometimes, it depends on the individual's genome thing. Personalized factors also plays an important role here. Some people will show up resistant to a particular drug in a very limited period of time, and some of them will in a 1 year or 1.5 years. So the drug also will become, most of the time it becomes resistant, and recurrence of the diseases most. So these are the challenging factors. Now coming to the research in our team is, we actually focus on all type of cancer, and today I'm going to talk about the research focusing on the glioblastoma. So based on all these research, the research on the different type of cancer cells, we specialize our research on glioblastoma, and the compounds that we have taken for our study is the phenol compounds. The phenols are, we know that it is a very good agent, chemotherapeutic agent to treat, it can induce apoptosis, it can cause stoppage of cell division. So a lot of good features are there. So it is considered to be a potential compound to inhibit the growth, further proliferation of the cancer cells. So we have three. I have given here three different compounds, which is like, it's very briefly mentioned over here. And if you are interested to know what it is, you can just visit the website of Molecular Signaling Lab of Tampere University to have the expansion of these compounds. And these are the references of my colleague who actually worked on this. And these are the three important phenolic compounds we have intensively studied. And based on the R-MIT, that is like the group that is present in the R, which is like either methyl or O-methyl, the IC50 value, or you can say its toxicity to a particular cancer cell varies. So overall study of the basic, generally we start with, I'll tell you like what is the workflow that we have done. But generally when we do in vitro analysis, the first thing and the foremost thing we did is like selection of compound, and then we will go for how these compounds really have impact or really induce apoptosis or cell death in that particular cell line, and the IC50 value. That's a very important point to understand. So out of this, we come for a conclusion that THTMP, this is a particular compound, is found to be more effective with the least IC50 value. So maybe I can explain you with respect to another. So coming to the next concept, which is on GPCR, which is a G protein coupled receptor. This is a receptor signaling pathway, and it's a very good target for most of the diseases, and especially multiple sclerosis or traumatic brain injury. So the right side, you can see the expression of RNA and the expression of the protein of this particular GPCR receptor. You can say as in specific, that is GPR17. GPCR is the biggest family, of which the GPR17 is one, comes under the GPCR. So we also want to focus on how this GPR17 influences like whether it overcomes the challenges what we have seen earlier. So we don't want to either stick on to only the compounds that really causes cell death. We also want to go with combining the other factors, like here we comes with GPR17. So as I said, the right side you can see the RNA expression as well the protein expression of the GPR17, of which the brain seems to be the toppest expression pattern. And in the left, I have given a simplest schematic representation of how the GPR17 signaling pathway is activated. So the toppest is like without any ligand binding, and the bottom one, which says that when a ligand binds, you can see a phosphorylation happening, which induces a lot of expression of other genes at the bottom that ultimately leads to the cell differentiation, right? So this is how the signaling happens with respect to GPR17. And we also want to understand the combination of THTMP, which is the toppest or the potential compound, and we also want to combine the ligand, which actually has the ability to bind with the GPR17 receptor. So combining both of these together as a combinatorial, what would be the impact on the cancer cell lines, especially on the GBM cell lines. So our hypothesis is surgery or radiation or chemotherapy, generally when all these protocols being followed, temozolomide, I just want to talk about temozolomide over here, which is the chemotherapy, common chemotherapy used for this kind of cancer. This temozolomide, in general, it also has impact on the normal cells, and so it has a very even though it kills brain cells, cancer cells, it also kills the normal cells. So we we're looking for the better options, always going for a better option which doesn't have any impact on the normal cells. So now the right side, you can see the right side of the slide where the phenolic compound and the combination of the GPR17 receptor, we also worked on a lot of compounds which actually targets the GPR17, and the top leading agonist, we call it as agonist, which actually gives a positive signaling processes upon binding with GPR17, and T0 is a tag of name or symbol we have given for the compound, which actually which is actually a potential ligand binding with the GPR17 receptor. So we propose, we plan to give in a combination of these two compounds, and we just want to see like what would happen with respect to the other molecular processes in the cancer cells, especially in the glioblastoma cells. So this is the workflow that is done to complete this particular hypothesis. And workflow, it starts with the docking simulation, which fits best those compounds were taken, and immunoblotting, other secondary messenger analysis, and its cytotoxicity, and have a lot of in vitro assays, which is a standard assays essential in order to analyze the efficacy of a particular compound, which is like migration. Like, it should not allow the cells to migrate. When you add a compound, the brain cells, or sorry, the cancer cells should not migrate, which is directly correlated with the concept that it stops the migration and invasion, and as well the colony forming, which is the ability to form the tumor. Like they can attach with the nearby cancer cells and develop into a tumor. So all those assays, and along with apoptosis, ability of your drug inducing the cell death, that was also been done. And we also want to understand how it really influences cell cycle pathways, which is the basic cell cycle pathway, where it actually influences either in S phase or M phase or in G phase, where it actually plays a role in stopping the cell cycle, and other free radical analysis like ROS, MMP, caspase. So these are the in vitro analysis. And apart from the in vitro analysis, the next stage of analysis like RNA sequencing, protein profiling. So these two stages of analysis will tell you what are the genes which really being influenced by the drug, and what are the genes which plays a vital role in particular cancer and in different stages of cancer. All those things can be analyzed by analyzing the RNA sequencing and proteomic profiling. And finally, every study involves a xenograft model. It can be authenticated when you do something. Even though you do a lot of experiments in cells, it has to be authenticated only once when you do it in the animal model. Then after successful treatment or successful experimentation or results in the animal model, it could be taken for the preclinical analysis. So this is what we have done. This is the workflow. So coming to I'm not going to show you the pictures of the graphs or mix of population of glial cells, cancer cells and non-cancer stem cells will be there. So this THTMP, the drug top leading compound, what we have identified. So what initially we did is we have separated those two cells from those microenvironment, and those two cells were individually treated with this particular compound THTMP. And it was also compared with the normal drug. It's very important that the drug, or it's very important that any kind of compound, you can say it as potential when it works better than the existing drug, right? So we also want to compare with the temozolomide variants. What is here given as a variance is we tried developing resistance ability for the cancer cells, brain cancer cells, to this particular temozolomide. And we also created sensitivity of cancer cells, sensitivity of the cells to this particular temozolomide, then the resistant cells and the sensitive cells, those two cells were treated with the THGMP. And even if it is sensitive or if it is resistant, this THGMP, which we proposed as a very top leading compound, it is able to sensitize, which means it is able to target any type of cells. This particular feature is mostly related with the factor what we see in the previous slide. Cells becomes resistant to the drugs. Okay, so when it becomes resistant, as I said, the treatment becomes failure. But when we tried with different sensitivity of the cells we created and we treated, it was very, very effective. And this compound was able to arrest the cell cycle at G1S as well in G2M, both the stages, and it is specific for different population. G1S for GSC, G2M for NSCC cells. So even for different populations, it was able to, according to the nature of the cells, it was able to stop, and it is able to induce apoptosis, cell death. We were able to identify that this compound was able to target EGFR as well the cancer stem cell signaling pathways also. The downstream signaling genes are there, and a lot of these genes are influenced in a positive manner by this particular compound, and so the induction of apoptosis was very, very successful. So whatever I have explained, I have correlated the features what is actually it is inhibiting. I have given it as a cross. And coming to the next combination effect, we have studied separately, and we want to, as I said, we want to combine these two compounds, and we want to check the combinatorial effect, whether it is working better than the individual THTMP or it is working good. And from the results, the combination, always a combination works better than the individual. Yes, of course, it showed a strong inhibitory effect with the least ICSFGD value, and it reduces all the migration, invasion, or colony formation, which are the special characteristic features of any type of cancer cells. And it arrests the cell cycle at S phase of the cell cycle. And other intracellular messengers like calcium and MMP, they were all increased, which actually leads to the process of the apoptosis and other molecular processes. And we were able to identify the pathway, what is the pathway it really takes up for induction of apoptosis. There is always an intrinsic and extrinsic apoptotic pathway. The pathway it takes is intrinsic pathway to kill the cells. And this we have taken it to the xenograft model. Xenograft GBM model was developed, and it was injected with this combination of the drugs, which showed a very equivalent, promising effect in reducing the tumor volume. As well, it showed increased survival rate of the animal when we given this combination of the drug. So when you see combination, too many circles, which is too many potential features, when you do it in a combination, could be achieved. And the final the work with respect to this particular objective is we also want to identify how the expression pattern also, when you add something, it does something, okay, and how actually it influence the expression. We could see that GPR17 expression is very high in the brain. From the previous slide I have said, so how this expression, endogenous expression, also helps in the survival as well in the treatment of GBM. And we first confirmed, yes, of course, there is an endogenous expression of GPR17, and we also found out that it showed higher cytotoxicity effect than the temozolomide existing compound. And this mediation of GPR17 activation, it happens through a lot of genes. I have given here as P13K act and MAPK, STAT, and NF-kappa B. So these are the major genes which is involved in the activation of the GPR17, and it was able to induce apoptosis and arrest cell cycle at G1. And very interesting factors, like it was able to even cross the blood-brain barrier, and we have gone for an additional examination here is like we have got the patient-derived GBM cells, and we were able to analyze the same data in the patient-derived cell lines as well in the xenograft model, which showed a promising results. So the expression also helps in giving the better results. So coming to the summary, from the previous slides, I've just made a gist of like a combination of these compounds was able to give or it increases the anti-tumor ability of this particular compound. This combination of the THTMP and GT0 compound. So the significance of our work is like we know that current drug, it doesn't shows efficacy on the heterogeneous group of population of cells, or it develops resistance to the drug, or recurrence is happening. And the solution that was like at present solution that is happening is like people are managing. We could say managing is like multiple targets. They were managing with temozolomide because it was able to target the multiple tumorigenic features and inhibits the multiple signaling pathway. But the exact, what we have identified through our research is like we were able to suggest that alkyl aminophenol might, along with GPR17, as a new target for GBM treatment, and the combination must be a promising candidate for GBM drug development. Thank you. So, yes, please. It's not like that. Actually, this drug was we did in all types of cancer, as I have given in the initial schematic representation. We worked with the breast cells, we worked with the colon cells, we worked with even liver cells, H329 cells. But intent, all the cells, it had its cytotoxic impact. But since we want to specialize our research in glioblastoma, we did very extensive work in this, and our collaborators are working on the other cancer cells also, which is giving very similar data. So it's not so specific to one type of cancer. This GPR17 is actually, this target is very a good target, and it's working for all type of cancer. So it's not as specific for GBM alone. Yeah. So the connection was there when I answered the question, right? Yeah. Yes. Yeah. So we were successful with the xenograft models